Okay, can somebody confirm you can hear me?
We can hear you.
Great, thank you. Let me share my screen here. Should be seeing my screen now. The Zoom meeting may close early, but usually with the lab, I'll just be talking for the first, I don't know, 40 minutes, half an hour, something like that. So it should be okay. If it does drop, I'll have to log in again. I don't know. Did you guys need to log in with the lecture or did it drop for you guys? Anyone?
It dropped for me.
Okay, so you had to log in again. All right. Um, I need to get the syllabus up. I know what we're doing. We're doing lab 2, but it's best to get the syllabus. Anyways, oh, not there. All right, today is October 2nd, and we are supposed to be covering lab 2, aseptic transfers. If you have not given me your infectious disease project, remember you need to okay it with me, and I will check it. There's been a lot of students who have had to narrow down the topic, and that's partly because we're a big class, so I simply narrow it down so that multiple people can work on one topic, like malaria. I can split it into the disease caused by 5 different parasites. Like the flu can be split into 3 different diseases. The flu caused by influenza A, B, and C. I don't think there's much difference between the flu caused by A, B, or C, but there might be some differences. I'll have to look that one up. Anyways, get it okayed by me, and then you can start working on the project. If you do not clear it from me, you will get a 10-point penalty. So you need to clear it with me first. And that's also to make sure that each individual has a different disease. All right, we're going to be doing lab 2, aseptic transfers today. Any question about any of that? Gonna close that down. Just my computer will work a little faster. I think we did finish the lecture file because it was a shorter file today because the lecture was broken down into 2 files today. All right, lab module 2, aseptic transfers. Upon completing this lab, you should 1, understand the purpose of using aseptic transfer techniques. 2, determine if aseptic transfer is used from the data provided. And 3, define the following terms. What is ubiquitous means? What is an aseptic transfer? What is an inoculation? And what is a contaminant? In our local environment, microorganisms are ubiquitous, meaning they are essentially everywhere. This is a problem when working in a microbiology lab because we generally want to work with a pure culture, a culture that contains one and only one species of organism. If you had multiple species in a tube and then the tube turned yellow, you wouldn't know if your species of interest, meaning the one species, caused the tube to turn yellow, or if some other species in the tube caused the tube to turn yellow. Louis Pasteur developed the technique of using aseptic transfer to minimize the risk of culture contamination. And Louis Pasteur also developed the technique of getting the first pure culture. What he did was he took a limiting dilution, meaning he made several different dilutions, and then replicated each dilution. So we would have 10 tubes to each dilution. And he had many different dilutions, 5, 6, maybe even 7 of them. And if ever he got a dilution where he got one tube that had growth and 9 tubes that had no growth, he correctly surmised that the one tube was likely to be a pure culture formed by one species. A high chance of it. It's not guaranteed. Nothing's guaranteed in science, but it was good enough for him to work with initially. That was actually a very difficult way of getting a pure culture. Edward Koch developed an easier way of getting a pure culture. And his way of getting a pure culture was he took mixed cells, streaked them on a solid surface like an agar plate to get isolated colonies. And he would take from one isolated colony on the first plate and then streak it out on the second plate to get isolated colonies. If he took from one isolated colony on the second plate and then grew that up, he would likely have a pure culture. It's not 100% guaranteed, but in over 95% of the cases, if you take from cells that have gone through 2 colony isolation events, you will get a pure culture in 95% or greater percentage of the time. A much easier way of getting a pure culture. Louis Pasteur then developed aseptic transfer to minimize the risk of contaminating your pure culture. And we'll learn about how to make aseptic transfer in this lab today. During inoculation, never lay your cap or the petri dish lid on the surface. So here we have a lid. I don't know if you can see that. Hopefully you can. My hand being the surface. You don't want to put the lid on the surface because the surface is not sterile anymore and it has microorganisms that have fallen out of the air or dropped onto the surface from maybe your use or maybe from you, depends. And you put the lid on the surface, it can pick up the microorganisms on the bottom of the lid. And now when you go to the lid to take the lid cover back, the microorganism can drop from the lid into the culture while you're putting the lid on, or it can drop when you take the lid off and you're moving the lid over the culture. You should especially do that with a cap of a test tube. Never lay it down because the tube obviously beforehand has been sterilized. So at least inside the cap, it should be sterile. And normally with the cap, you hold it with your pinky in the hand that's holding the loop, and then you hold the other tube with the other hand. And when you're working with the tube, you hold it at an angle. So here, this is kind of a full glass here of water. I don't know if you can see the water or not, but pretend that this yellow end of the pen is the loop. You hold it at an angle, and I can't hold it at a very steep angle because the water will drop out. And then put the loop in. And that way, the surface area that's open will be reduced because this is a large surface area. Let me drink some of the water here. So now I can hold it at a steeper angle. Still can't hold it too steep, but you can see that now if the something drops down here, that's fine because that's the outside of the tube, and it would have to go exactly in this region here, which is less than this region here. Hopefully you can see that. Could you see that? Anyone? I tried to have it in front of my face so you could see it. I'm sharing my screen so I can't see it, but could you guys see it? Nobody's going to answer. All right, we'll move forward. Anyways, you want to hold the tube at an angle, hold the lid of the tube with one hand, the loop with one hand, and that way there will be less contamination from the air. You still are at risk for contamination as long as the lid is off the tube. And then you want to do this quickly and then quickly cover the tube with the cap so that once that happens, there will be no more risk of contamination from the air. And you then have as little time with the tube open as possible to reduce the contamination by the air. For the Petri dish, what you do is you obviously have the lid open for as little time as possible, do your thing, and then obviously don't put the lid down on the surface. And actually, with the lid of a Petri dish, you keep the lid over the dish like this, and then stick the loop inside. And it's better maybe to use it right here. And this way, if something falls down, it's most likely going to hit the lid, and that's okay because the top of the lid isn't sterile anyways. It prevents the bacteria from falling out of the air and into the dish. You would have to have the bacteria exactly fall in this region here, which is open, to contaminate the dish. And most of the dish is protected by the lid. And then, like I said, keep the lid, have the lid off the Petri dish for as little time as possible. And that will reduce contamination of the tube or the dish from the air. All right, so making a septic transfer, you must assume everything is contaminated. Even the surface of the desk that you cleaned, something can fall out of the air, but you obviously want to clear the surface of contaminants. So you initially work on a clean surface, but with time, obviously, the surface will start getting microbes on it. And then you sterilize everything else, like the loop, the tubes you use. Obviously, you can't have the stock tube sterile, but the sterile medium should be sterile before you use it. And then how you sterilize the loop is you put it in the flame or the back to incinerator until the wire portion of the loop is red hot. And then you remove the loop from the back to incinerator or the flame. You need to cool it. You can cool it by holding it in the air for 20 to 30 seconds. That's not the best way to cool it because the air, you could risk having bacteria fall out of the air onto the loop after it's cool. Because the loop is now cool, it won't kill the microbe. And obviously, if you do that, you want to use the loop right away so there's less risk of something falling out of the air and onto the loop. A better way of cooling the loop is you put it in the back to incinerator until it's red hot. You take it out and then you put it in the stock culture, touching the side inside the glass tube of the tube. The inside is going to be sterile except for the microorganism in the tube. Cool the loop on the side of the tube, then pick up your microbe from the stock culture and then quickly transfer it into the sterile medium, having the tube open for as little time as possible. Any question about any of that? And that's aseptic technique, having everything sterile that could contaminate the sterile medium. Have everything sterile so it doesn't contaminate the sterile medium. Obviously, you have to take the lid off, so there always is a risk that something could fall in from the air. But you then hold the tube at an angle so that there's less risk of something falling out of the air. All right, know the terms of this lab? In this lab, you're not going to be physically performing any of these activities, but you will be watching videos of these activities, and we expect that you'll know and understand the material in these videos and in this lab manual. In procedure 1, you're going to transfer sterile liquid, water, using a glass serological pipette, putting 1 ml of water into 9 ml of sterile media. That's going to have a problem, and that is when you transfer sterile water, either in a biuret or in a pipette, even a plastic pipette, the water will not form flatly against the surface. Especially if it's glass, you'll have quite a big meniscus where the water will attach to the edge more than it will in the center of the meniscus. And where do you read the meniscus? Do you read it at the top? Do you take the average, meaning it's someplace in between? Or do you read the meniscus at the bottom? We always read the meniscus by the bottom of the meniscus. Any question about that? That is the tube that we will inoculate with the sterile 1 ml of water. And you can see the media in there and whether it's cloudy or not cloudy. And then use this table for the interpretation of your results and conclusion. If the result is that the tube, the broth is clear for procedures 1 and 2, then the conclusion is no organisms are growing in the broth and aseptic transfer was successful. If the nutrient broth is turbid, hazy, or cloudy, then organisms are growing in the broth and aseptic transfer was unsuccessful. In the questions for procedure 1, it asked in 1B, based on the results, what conclusion can be drawn? You must use wording similar to the table, where you're talking about whether organisms are growing in the broth or not, and then whether aseptic transfer was successful or whether it was unsuccessful. So you must use either wording similar to this table or exactly as this table states it, or else the question, that one that I just showed you, will be at least partly wrong. Any question about any of that? All right. For procedure 1, here is the tube after we've inoculated the media with 1 ml of sterile water, and then we incubate the tube in the incubator overnight, and then we take a look at the tube. And then answer the questions for 1, obviously answer the question 1, what's the initial clarity of the nutrient broth prior to the inoculating, adding sterile water or the paper disc? And was it clear or was it turbid? You can take a look at the picture there. And let's go ahead and go to the worksheet now, because we're in the questions. And then for procedure 1, answer question 1. Describe the clarity of the nutrient broth after inoculating with sterile water and overnight incubation. Was the tube clear or turbid? So write clear or turbid. And then 1B, based on these results, what conclusion can be drawn? Your conclusion must at least use words similar to or exactly in the table above, or else it'll be partly wrong. Or completely wrong, depending on what you answer. All right, list 3 sources of error with aseptic transfer technique that could explain why the nutrient broth showed growth of microbes. So 3 potential errors. Give me 3 potential errors for 1C. Any question about procedure 1? We choose 2P1, question 1. All right, if not, let's move on to procedure 2. Procedure 2, you're going to use a septic transfer technique to transfer a sterile paper disc into nutrient broth. Let me switch glasses here because you'll see these better, I think. I don't have not a scalpel, what the heck is that? A forceps to take the paper disc out of the sterile petri dish. So the paper discs are sterile, but use forceps. You cannot get the forceps red hot because if these were the forceps and you were to get the tip of the forceps red hot by putting it in the flame or the back to incinerator, the heat will move up the forceps and then you could not handle the forceps. So what we do with the forceps is we stick them in alcohol, getting the forceps wet in alcohol, and then you put the forceps either in the flame or in the back to incinerator. And I should say you only put the tip, the far side tip of the forceps in the alcohol, and then you put it in the back to incinerator, the flame, to burn off the alcohol. Now, the forceps are not sterile, but they're clean. There's very few microorganisms that can survive the alcohol and then the flaming of the alcohol. And then you go and you pick up the paper disc, and I don't have a paper disc here, so hopefully you can see this. We're going to pretend that this, grab this here. And I know you can't see it. I'm trying to grab it before I hold it because it's just too difficult to hold the paper disc in the, there. Hopefully you can see that now. And I'm going to lift the paper disc out of, just dropped this, sorry. Ah, the glasses pretending to be the forceps just aren't working well. Maybe I should use my fingers, then I could grab it easily. Come on, come on. Oh, there we go. Hopefully you can see that. We're pretending that lid is, it's a lid to a computer desk, is, uh, or the top is the paper disc, and then you pick it up and you put it into the sterile media. Any question about any of that? Put on the lid to the media, to the tube, and then put the tube in the incubator and incubate it overnight. And that's procedure 2. You then use the table once again for procedures 1 and 2 for getting the results. If the nutrient broth is clear, then no organisms are growing in the broth and aseptic transfer was successful. If the nutrient broth is turbid, hazy, or cloudy, then organisms are growing in the broth and aseptic transfer was unsuccessful. And then you look down at tube P2 for procedure 2, you can actually see the paper disc in the bottom of the tube there. And then answer the question. 2A, was the clarity of the nutrient broth and the sterile paper disc tube incubated overnight, was it clear or turbid? So give an answer there. And then based on these results, what conclusion can be drawn? Remember, your conclusion must use wording similar to the table or above, or it could be the exact wording as the table above. Any question about procedure 2? If not, let's move on to procedure 3. Procedure 3, you're going to transfer Serratia marcescens to a broth culture. Serratia marcescens is a good organism to transfer because it is colored, it is pigmented. We have a video for you to watch for procedure 3 and 4, demonstrating how to correctly transfer from a bacterial stock culture to a sterile broth and a sterile agar slant. So watch that video. You should note that there is an error in the video, and you only need to watch the video for up until 4 minutes and 23 seconds. After that, you can stop watching the video. The video is transferring using bacteria E. coli, which is different than our bacteria. E. coli is not pigmented. Our bacteria is pigmented. And there's an error in the video when the instructor puts the loop into the tube of E. coli between 3 minutes and 5 seconds and 3 minutes and 20 seconds, you can hear something that is the error. You can hear sizzling when he puts the loop into the E. coli. Can anyone tell me what the error was? Anyone know?
He didn't cool the loop down before putting it in the E. coli.
Correct. That was his error. You should cool the loop down because if he did not cool the loop and he sticks the loop into the bacteria, if the loop is still hot, especially if it's red hot, it can kill all the bacteria. So you want to cool the loop down first and then pick up the E. coli. He didn't do that. He picked up the E. coli and apparently had a liquid culture. And so probably, as long as he swirled the loop through the liquid culture, he'll probably cool the loop in the liquid culture and then pick up bacteria. But don't do that because if you are picking up cells from a solid culture or you put the loop in the bacteria very quickly and then pull out the bacteria, the loop can still be hot and kill off the bacteria. All right, so procedure 3, we're going to transfer Serratia marcescens to a sterile broth culture. Remember, that's a liquid culture. Serratia marcescens is pink when it is grown in a liquid culture. It's red when you grow it in a solid culture, but it's pink when you grow it in a broth culture. Broth cultures can be useful, but there is a problem with the broth culture, and that is all the cells grow together in the broth, and you cannot see individual colonies in the broth, which I guess I can show you down here. You can't see any colonies in that tube. And so if you have slight contamination, the tube will still be pink because Serratia marcescens is the majority of the organisms in there, but you'll have a few contaminants in there. And even though they're not pink, they're some other color or they're non-pigmented, you'll still see the tube being pink from the majority Serratia marcescens. And that's the problem of broth culture, and that is you cannot be certain you don't have a minor contaminant in the broth culture. Any question about any of that? All right, and for procedure 3, use the conclusions come from these results. If the nutrient broth is clear, then no organisms are growing in the broth and aseptic transfer was unsuccessful. If the nutrient broth is turbid, hazy, or cloudy, then organisms are growing in the broth and aseptic transfer was successful. My question is for procedure 3, if the nutrient broth is turbid, aseptic transfer was successful, and yet for procedure 1 and 2, if the nutrient broth is turbid, hazy, or cloudy, then organisms are growing in the broth and aseptic transfer was unsuccessful. So why is there a change? Why in procedure 1 and 2, if it's turbid, then the aseptic transfer was unsuccessful, but for procedure 3, if the nutrient broth is turbid, why is the aseptic transfer now successful? Anyone know?
Because one, you're moving a medium, one and two, you're moving a medium into the tube and agar plate, or in the second one, you're moving actual organisms, so you want there to be growth?
Yes, in procedure 3, we are actually transferring cells into the tube. And so we expect cells to grow into this tube. So aseptic transfer will be successful if it's turbid. If there's no turbidity, there's no organisms growing, and we didn't transfer anything. So aseptic transfer was not successful. However, in procedures 1 and 2, we're transferring only something that's sterile into the tube. So we expect that the nutrient broth will be clear. Because if aseptic transfer is successful, there should be no organisms growing in the tube. Anyways, the answer was correct, so let's move on. The questions, there's the tube for procedure 3, and you can see it's pinkish. And then answer question 3. The results for P3 describe the clarity of the nutrient broth after inoculating with Serratia mersens and overnight incubation. Was the tube clear or turbid? So answer that for 3A. And then 3B, based on the results, what conclusion can be drawn? Once again, your conclusion must use wording the similar to or exactly the same as in the table above, or else it will be partly wrong. And then for procedure 3C, or question 3C, how do you know whether you've transferred the correct microbe, Serratia mersens, or not? There's an easy way to know for this. And then for procedure 3D, if there is only slight contamination in the broth, such that Serratia mersens was the main bacteria present, but there's slight contamination, explain why most likely you would not be able to detect the contamination. All right, any question about procedure 3? All right, let's move on to procedure 4. I need to go quickly through this because this lab is going to end shortly, the Zoom meeting. And I can restart it, but I'd rather not. Using aseptic technique, transfer Serratia mersens into a sterile agar slant. When Serratia mersens grows on a solid surface, it will be pigmented red. So we have a sterile tube of agar, and there's one there, and you actually streak the Serratia mersens in an S-like fashion along the slant there. And then when it grows up, if you have a contaminant growing off the slant, let's say we had a colony growing there off the slant, then you know that's a contaminant. Also, if it's growing on the slant and it's a different color than red, you also know that it's a contaminant because Serratia mersens should be red. Any question about any of that? It's much easier to detect a contaminant on a solid medium than in a broth. The conclusion, use for the results, if the slant does not show confluent or colony growth, then no organisms are growing on the slant and aseptic transfer was unsuccessful. If the slant does show confluent or colony growth on the slant, then organisms growing on the slant and aseptic transfer was successful. Here we are seeing procedure 4, and you cannot even see the streak because there's so much growth that it's just confluent wherever we did the streak, although you can see a little bit right here and a little bit right there where there is no growth because we didn't streak over here and didn't streak over there. Down here, there's a little bit of water here, and because of the water, the bacteria moved all the way across. Okay, and that's why it's a little bit lighter there. It's just because of the water still surviving in the tube and it's down there. Anyways, there's nothing growing here, nothing growing there where we did not streak, and there's no colony that's not reddish there. So it looks like this is a pure culture. And then answer the questions 4. Is there bacterial growth on the slant? Answer yes or no. And does the growth appear to be the bacterium you inoculated or a contaminant? and give an explanation for why. All right, any questions about procedure 4 or any questions about the lab? If not, I will leave it here. I will log in case somebody has a question, but once the last student has logged out, I'm going to log off. And I think this is going to continue only for about 2 minutes before the Zoom stops. My apologies about that. Zoom isn't working correctly.
Thank you. Good night.
All right, go ahead and get started on the lab.